forgot to record this. So that's direct contact. Animal cells also can use a cell-to-cell recognition system, which again, I think you guys are probably more familiar with than most people are, because at some point in your life, you've probably taken a COVID test, which is an antigen test. So you've heard the word antigen over and over and over and over and over, whether you know what it is or not, you've at least heard the word antigen. This is a way that cells can use surface molecules to connect and share information. So this cell here has a receptor, and this cell has another receptor, and those receptors can bind, and that causes some kind of signal to go out. So when this binding happens, that causes a signal to go out. So this is using cell surface molecules, like we talked about when we talked about the cell membrane. These could be glycoproteins. So proteins in the lipid bilayer that have carbohydrates attached to them. Those carbohydrates can give information. These can also be glycolipids. Lipid molecules that have carbohydrates attached to them. So this is really important in things like the immune response. So when something that is not belonging to the body comes in, it has antigens on it that are like, your body recognizes and says, hey, you're not one of us, and so we're going to get rid of you. So it sends out signals to other immune cells to attack, to get rid of, to destroy, and to remember them for the next time so that this doesn't happen again. It's also really important in embryonic development. But again, that's considered direct contact because the cells are actually connecting with each other. Most of this chapter, we're going to be talking about ways that cells communicate indirectly. These other two on this slide are also they're not direct, but these are ways that it doesn't involve direct contact, but not like super complicated yet. So there are ways that cells can signal locally. So this is being able to signal cells that are only a few cells away. So this works in cells that are in close proximity. Cells that are close to each other can signal each other using chemicals. So we have a cell here, and this first example is what's called paracrine signaling. So you have some kind of cell that is called the signaling cell that needs to send a message out. And it's going to send that message out molecularly, so chemically. Do humans communicate chemically? Pheromones are chemical signals that humans and animals can use. So but again, this is only talking about a few cells away. This isn't massive communication. So this signaling cell is going to send out some kind of signal, which is going to be able to hit specific target cells. Again, only cells that are pretty close by. Some examples of this molecules that might, or messages that might be sent out are growth factors, cell division, molecules that would signal a cell to divide. And again, a lot of regulation in embryonic development. So just a small amount of ability to communicate. This would only being able to communicate a short distance, but important things that can be sent out that way. So that's one kind. A second kind of local signaling is synaptic signaling. When we talk about synapses, or if you look at this picture, does this look familiar to you at all? Like where in the body might this be happening? Nerves. This is the nervous system. So you've got the axon of one nerve interacting with the next cell. So this is occurring in the nervous system. So this is only happening in animals that have a nervous system that's developed enough. Plants can't do this. In this case, there's both electrical and chemical communication going on. So the message is coming down the nerve as an electrical signal. So this electrical signal comes down, and that's going to trigger the release of neurotransmitters, which are chemicals. So the electrical signal is coming down. It's going to trigger the release of these neurochemicals, these neurotransmitters, which are going to diffuse across the synapse. So this gap is the synapse. To this cell, which is the target cell. This can happen over long distances because you have really long nerve cells, but it's still only dealing with like one cell to one cell. So paracrine signaling is using a chemical signal, and synaptic signaling is using both electrical and chemical signals to send a message. Like, hey, move, muscle. When we talk about long distance signaling, this is where we're talking about endocrine signaling. What do we mean by endocrine? Yep, this is hormones. So this is longer distance messages, but still chemical messages. So they're long distance chemical messages. And again, these are going to go into the bloodstream, but they're only going to affect certain target cells. Because only those target cells are going to have the right receptors for that hormone to be able to bind so that it can have its effect. Okay, you don't want to send a message to the wrong kind of cell. You want to make sure the message is going to the right kind of cell, so the message has to bind to the right kind of receptor. When we used to pass notes in class, you don't want the note to go to the wrong person. If you had any life experiences with notes, and you can, anybody write notes on paper? You know, you're passing a note and somebody else opens it and reads it. That's not what you want to happen. Had a specific recipient, so you would write "for your eyes only" on it, and then you would know that only that person would read it. Okay, plants and animals both have hormones. So when we talk about the bloodstream, obviously plants don't have a bloodstream. Plants diffuse hormones just through simple diffusion, and they can move it dissolved in water through the xylem and phloem. But plants and animals both do have hormones. So we're pretty familiar, at least with some hormones of humans and animals maybe. Does anyone know any plant hormones? No, because nobody loves plants. Yeah. Ethylene is a plant hormone that is used to ripen fruit. So a lot of fruit that is picked unripe to be shipped, because we're shipping fruits over long distances, and bananas, you don't want an overripe banana. So a lot of fruits are picked not ripe, shipped, and then treated with ethylene. Ethylene is actually a gaseous hormone. It's small enough that it can be a gas. And so you can spray ethylene gas onto a fruit and make it ripen. I've heard, I don't know if this is really true, but I've heard that they pick oranges like totally green. And then they ship them, and then they ripen them, and they are orange. I don't know if that's true. I've also heard this is a total side note, but the color of the mesh bag that they put fruit in affects your perception of the color. The oranges are always sold in like orange or red bags, and limes are always in green bags, and lemons are always in yellow bags because it makes them look more ripe and makes them look better. Avocados are sometimes in purple bags, and I don't know how that makes you feel about avocados, but anyway. So if you ever had bananas, bananas produce a lot of ethylene on their own. So if you put a banana and another fruit together, the banana will produce enough ethylene to make other fruits ripen. There's other plant hormones like cytokinins that help with root development and auxins that help with shoot development. If you've ever put a plant in a window and it starts to grow towards the light, that's because the plant is producing hormones that make the cells on one side grow faster than the other cells, so that the plant will bend towards the light. So plants don't have the same kind of quick response with hormones necessarily, but they're still making hormones, and I don't want to leave them out of the hormone party. Okay, so that's kind of direct contact and simple methods. This is where we're going to get into the harder stuff, which is the ways that signal transduction happens, which signal transduction is actually, I don't really like that there. So when we talk about cell signaling, there are 3 stages to the cell signal. Okay, there are 3 ways that cell signaling happens. 3 steps of cell signaling, 3 stages of cell signaling. So the first step is here. This is signal reception. And that is what it sounds like. The signal has to be received. Okay, so in order for that to happen, we have to have a signal. So there's some kind of signaling molecule that is in the extracellular fluid. And we're talking about this very generically. Again, thinking about fight or flight. When we talk about the fight or flight in adrenaline, adrenaline target cells usually targets muscle cells and liver cells, which makes sense in terms of muscle cells. Why would it target liver cells? Nobody really knows what a liver does. You have it, it's big, it's in there. One of the jobs of the liver is to store glycogen. Okay, so glycogen is storing a way to store that glucose. So when we need to release glycogen, it's really great if the liver can do that because that's where the glycogen is. So there has to be a signal sent out and there has to be a receptor for that signal. So in the case of adrenaline, it's either muscle cells or liver cells are the main ones. So the signal is received. Then you see there's a pathway here. So the signal comes in, binds with this receptor, and that triggers this pathway, metabolic pathway. And that's what we call signal transduction. So you'll see there's 3 molecules involved. Sometimes it's only 1, sometimes it can be more than 3, but there's a pathway of moving that signal from the receptor to the target that's going to cause the response. So this is called a signal transduction pathway. And again, it could be 1 molecule, it could be 3 molecules, it could be 15 molecules. But this is what's carrying the message. So this is carrying the message to where it needs to go. This to me looks like the game of telephone. I contemplated trying to play telephone, but there are too many of you. But if you've ever played telephone, does the message get where it's supposed to get? Usually in the right form? No. Your cells are better at this form of communication than you are, thankfully. Okay, if we were dependent on our ability to telephone messages, we would be in trouble. But the signal transduction pathway is a pretty set pathway, and it usually is successful. And it's going to cause some kind of cellular response. So we have a signal being received. We have the signal being moved to where it needs to go to the target so that we can cause some kind of response. And this is a specific reaction. It's going to activate some kind of response in the cell. That cell may be, that message might be, take this enzyme, use this enzyme to break down glycogen. It might be, hey, we need this part of DNA transcribed and translated. So it can be any kind of message and it's going to trigger some kind of response. I keep seeing teaching analogies in this. Like sometimes I feel like I'm sending out signaling molecules and some of the receptors aren't binding. Sometimes the receptors are binding, but there's no response. I don't know. It's Friday. You just came from a test. So that's kind of an overview. We're going to get a little bit specific and talk about different kinds of signal transduction. I'm not anticipating that we will get through all of these today. I'm hoping we get through these too. We'll see. Okay, so there's specific kinds of signal reception, specific kinds of receptors that fall into categories that we will talk about these examples. So today, the first one we're going to talk about is a G protein coupled receptor, a GPCR. So again, thinking about reception as the message goes out, but only specific cells will receive it. This signaling molecule, whatever the signaling molecule is complementary in shape to the receptor. Complementary. Which might remind you of something else that we talked about. What else did we talk about where the shape and there was specificity for binding? Enzymes, yeah. Enzymes work with a specific substrate, just like these signaling molecules work with a specific receptor. So they're not going to bind to the wrong thing. We also call signaling molecules, and I didn't write this very well, ligands or ligands. It depends on who you're talking to. Ligand. That's referring to a small molecule that will bind to a larger molecule. So signaling molecules are ligands, not ligers, ligands. So a ligand is anything that specifically binds to another molecule. In this case, usually it's something that's bigger. When this bonds, when the ligand bonds, it changes the shape. This is kind of generic signal reception. We're going to get to the GPCRs in a minute. So that binding triggers some kind of change of shape in the receptor, which is a protein. And that change in the protein is going to activate it. So when it activates it, that allows it to interact differently with some kind of molecule on the inside of the cell. To cause that reaction. So there are, on the surface of a cell, there are three major kinds of cell surface transmembrane proteins. What do we mean by transmembrane? It goes all the way through the membrane. So that's what you're seeing here. So it's touching both the extracellular surface and the intracellular surface. And here, what is this protein here? It's not transmembrane. It's called what? It's on the side. Starts with the P. It's a peripheral protein. So these are transmembrane proteins or integral proteins, and this is a peripheral protein. So one of the kinds of cell surface transmembrane proteins that we're going to talk about, the three major types. The first one is the G protein coupled receptor, the GPCR. So this is the actual GPCR. On this side of the GPCR, so on the extracellular side, it's going to have, oh, I thought I changed the color. Some kind of binding site. So that binding site is specific to the signaling molecule that's going to come in. On this side, it's going to have some kind of site that's going to interact with the protein, with the G protein. In your body, there are, well, not in your body. We know of over 800 different kinds of GPCRs. So there are ones in your body, there are ones, yeast cells have them. Neurotransmitters, epinephrine, and other hormones are G protein coupled receptors. Vision, taste, and smell, those senses are controlled by these GPCRs. And again, a lot of things in embryonic development. They are all made up of a single polypeptide. Polypeptide is a chain of what? Chain of amino acids held together by what kind of bond? Peptide bonds. So that's, the chain is our primary structure. What's the secondary structure of proteins? They can do what? They can twist into helixes, or they can fold into pleated sheets. So you can have alpha helixes or beta pleated sheets. So this is a single polypeptide. It's made up of 7 different alpha helixes. So it doesn't have that quaternary structure where we put a bunch of polypeptides together. So in this first step, we've got our GPCR with our binding site, a site that's able to interact with the G protein. And this guy here is it's an inactive one. It's an inactive G protein. Why do you think it's called a G protein? What's bound to it? What? It might be functioning as an inhibitor. Specifically, it is GDP. What do you think GDP stands for? Let's start with ADP. What does ADP stand for? Adenosine diphosphate. This is a similar kind of nucleotide. It's guanine diphosphate. Okay, so it's very similar to ATP. It's just got a G on it instead of an A. And it has 2 phosphate groups. It's inactive when it's GDP. So when GDP is bound, this is what's making it inactive, is that it's GDP. And this over here, this blue thing, is an enzyme. This is our target. This is what we're trying to get to do something. So that's the setup. Step 2 is showing you what can happen. So we've got our signaling molecule coming in. And it's going to bind to that receptor site. What is that going to do? When a signaling molecule binds, what does it do? It's going to change the shape of that protein. So it's going to change the shape of that GPCR. And that's going to make it activated. So it's now activated. Okay, now we're going to bind GTP. So this activation causes the G protein to bind, GTP. So this is now activated. And we know it's bound because now it has GTP. But still nothing has happened to the enzyme. Okay, in step 3, the G protein is going to dissociate from the GPCR. Sorry, I thought I picked the right color. So the G protein dissociates. From the receptor. And then it actually diffuses down the membrane. I don't have enough space. And it's going to bind with the enzyme. So now that's going to activate the enzyme. So once the enzyme is activated, that's going to cause some kind of cellular response. Again, we're talking about this very generically. At this point, the signaling molecule can dissociate. I'm not spelling that right. We're just going to say releases because I can't spell. So the signal molecule is released because this binding is reversible. It's not a permanent binding. So this is going to release. Once that is released, it can go back and do this again. So it's reusable. This enzyme is this GTP is going to release a phosphate group to be GDP to inactivate the G protein. Because oh, I don't like it. It's going to trigger GTPase. GTPase is an enzyme that can remove a phosphate group from GTP to make it GDP to inactivate the reaction. So we don't necessarily want the reaction to keep going on and on and on. So this is a way to stop this signal transduction pathway is by activating the GTPase to make this GDP again to inactivate it. So that will stop the pathway unless there's another signaling molecule present, in which case it will restart the pathway. So it's self-limiting in that if there's no more signal molecule, it will stop the signal from going out. So it'll stop the response from happening. Because your cells don't want to keep responding to a situation if there isn't really a situation to keep responding to. We are obviously not going to get through the next one. So we are not going to start receptor tyrosine kinase. What are you going to do at 11:30? You're going to come celebrate all day.